swine it. It's time for a new era of communication in the swine industry, one that you can get the latest updates while commuting or driving to farms. Here you have the brightest minds of the global swine industry in your pocket.
Always take a look at the microbiome, see what the microbiome reveals. I would predict, Marcio, that in the future, in 10 years from now, microbiome is going to become PCR. That means every lab in any university or any lab in any company that makes science or that makes feedstuffs or any other kind of feed additive is going to have a microbiome station.
Welcome to Swine Eat Podcast. My name is Marcio Gonçalves, your host for today's episode. This episode is brought to you by Every Pig. The truth is, precision swine production is not the future, it is the present. Every Pig is the intelligent pig health platform. It is a simple yet powerful pig health and production management tool. Request a free 20 minutes demonstration at www.everypig.co. Hello everyone, today we're going to talk about microbiome, teasing out the buzz from the reality. Today we have Dr. Andres Gomes from the University of Minnesota. And Andres, thanks for joining us today.
Thank you. Thanks for inviting me. It's a pleasure for me to be here.
Appreciate that. So Andres, just to start things off, if you can walk us through how did you get involved in pig production and talk a little bit about your career until today.
Sure. So I'm basically a microbial ecologist. That's my training. That was my training during my master's and PhD. And my main interest is to investigate and explore the composition and function of microbes in different systems. So I started my career studying soil microbes or microbes living in soil at different depths.
Interesting.
Then I I moved on to looking at the microbiomes of wild primates, specifically gorilla populations in Africa and also human populations in the Central African Republic, just as a way to understand the evolution of the human microbiome and how diverse lifestyles have altered the microbiome to the point it's become a little bit different from what our ancestors were supposed to have. So that was a very important topic in my research. It still is. But now as a faculty member at the University of Minnesota, I'm interested in the microbiome of food animals, and I have a very important research portfolio on the microbiome of swine. Basically exploring the swine microbiome and more importantly, trying to look at strategies to manipulate it to improve health and performance in pig production systems.
Very nice. That's a very diverse background there, from gorillas in Africa to pigs in Minnesota. I love that.
Yeah. I mean, but if you think about it, it's not that different. I mean,
Right.
We can talk about it later in more detail, but the systems themselves, as far as the capacity to metabolize different substrates in diet, is not very different from a gorilla, or comparing a gorilla to a pig, actually.
I can imagine that. Very nice. Well, since we are at it, Dr. Gomez, if you can tell us, what is the microbiome?
Well, the microbiome is basically defined as the collection of microorganisms that colonize or inhabit different surfaces, either within animals or in the environment. So all the collection of cells that are or that represent different microbes, even bacteria, fungi, viruses, and protozoa, is collectively known as the microbiome, along with the functions they encode. So it's not just the cells of the microbes themselves, but also the functions, the metabolic capabilities that they have. So that's what we define as the microbiome. So the microbiome is not just confined to the gut. We have a microbiome also in the nasal surfaces, in the skin, in the reproductive tract, and anywhere you look at, we're just surrounded by microbes. This is a microbial world. That's why the area of microbiomes is so exciting today.
Very nice. Well, on that note, why is there a hype about gut microbes in different areas of science?
Well, the hype basically, I would say, so microbial ecology is a very old field. If you think about what Anton van Leeuwenhoek started doing with the first trials of the microscope and these little animalcules he saw in teeth scraping, he was doing microbial ecology. He was basically looking at different microorganisms in teeth surfaces. So those were the initial, let's say, the initial steps of microbial ecology until what we know it, or as we know it today. And I think what revolutionized the microbial ecology or the microbiome field was the implementation of molecular tools to study the microbiome at high resolution and in many, many different subsamples simultaneously. So with the implementation of next generation sequencing, we realized that the diversity of the microbiome in different systems, especially in the gut, in the human or animal gut, is incredibly high. So when we started, applying those molecular techniques to study microbiomes in different systems, we realized first that the microbiome is more diverse than we initially thought, and second, that the functions that the microbiome performs or encodes are essential for life as we know it, right? So all the functions the microbiome performs as far as nutrient processing, nutrient synthesis, the equilibrium between the microbiome and the immune system are functions that guarantee life as we know it. And without the microbiome, those functions would not be possible. So in other words, there's functions performed by the microbiome that guarantee life as we know it. And without these functions, we wouldn't be able to exist. So I think that's part of the reason, or that's the main reason actually, there's such hype about the microbiome. That we realize based on all these new molecular and sequencing techniques that we use to study microbes, we have realized that the functions that the microbiomes perform extend the physiological capacities of animals and humans in a way that will guarantee life. And without those functions, we wouldn't be able to exist.
Interesting. Yeah, there's definitely has been an explosion in microbiome research in the last few years, right?
Yeah. And I think most of this hype began with the work that Jeff Gordon of University of Washington started doing with, I don't know if you know, this is a quintessential paper on microbial ecology about transferring the microbiome from an obese mice to a mice that is lean, and looking at how the phenotype of obesity is transferred along with the transfer of microbes. So that shows that there are some functions performed by microbes, as for example, energy harvest, that can actually be transferred from one organism to another. So that's pretty amazing. So from those trials and those experiments performed by Jeffrey Gordon, then we realized that the functions that the microbiomes perform, not only at the gut level, but also the gut-brain axis. There's constant communication between microbes in your gut and your brain, and we believe that there's an association between certain behavioral traits and the function and the composition of microbes in the gut. So that's another area that is receiving increasing attention today. Also, the microbiome role in different diseases, for instance, in cancer. We know that there is a connection between the presence and the function of certain microbes and the developing of certain diseases, as far as cancer, diabetes, autoimmune disease. So that's part of the big hype that we have about the microbiome today, and the expectation that we can change things in the microbiome to alter phenotypes, which we haven't been able to completely do, but there's a lot of promise on that. So when you think about microbes and the genetic makeup that you have, we're able to predict the probability of disease with higher confidence by looking at microbes compared to when we look at genomic regions in our own genome. So, some people are predisposed to certain diseases because they have a mutation or they have a SNP in their genomes that is associated with that disease. But we realize that the predictive power of the genes, of the genomic makeup, is not as strong as the predictive power of the microbiome when it comes to predicting disease outcomes.
Wow, that's amazing. Yeah. Well, so on that note as well, what is the state, Andres, of the gut microbiome research in pig production, and what can we conclude so far?
Well, there's been also an explosion of different work on analyzing and exploring the swine microbiome. The first work just intended to look at that microbial ecology, especially along different segments of the GI, when you compare small intestine, large intestine, feces, what is the microbial ecology on those specific segments? So the first work really intended to look at what is the composition of that microbiome, just to get a grasp of the microbial makeup and the functions they perform. So apart from that, then we've been moving from those, let's say, compositional surveys to specifically explore the function of those microbiomes, what kind of functions those microbiomes are performing. And then we've been moving to actually exploring manipulation of those microbiomes to see if performance can be improved. So there's a lot of work being done on prebiotics, in probiotics as well, in different organic acids and other feed additives that we know have some influence on changing microbial composition. But the manipulation of the microbiome as far as, feeding, let's say, prebiotics and probiotics and looking at that response in the animal is not always reproducible, which means we know there's a microbial ecology that is very specific along the GI of the pig. We know that by incorporating different additives in feed, we're able to get some changes in the microbiome, but we're not always able to get a response as far as the phenotype we want to reproduce. And the reason is that the microbiome in pig systems is highly variable across different individuals. So different individuals have very different microbiomes, even within the same species, the basic commercial, the white pig. When you look at the different microbiomes in different subjects, we see that the microbiome is highly variable. So that's one of the reasons why we haven't been able to replicate or to specifically manipulate microbes to improve specific phenotypes in pigs. So I think that's the state of the art of the pig microbiome work today. First of all, looking at looking at the composition and the function of the microbiome along different segments of the GI tract, and then trying to manipulate those microbial communities using different additives to see if we can improve performance and alter the phenotypes of those pigs. Except that these responses are not always constant and reproducible.
Highly variable. Interesting. One thing that you and I talked in the past, I believe, was a lot of literature is inferring association, right? And there is some recent work that is doing more of a randomized type of approach, even in that, fecal transplantation, all that kind of thing. So can you give a comment to us and to our audience about, the work done in microbiome on association and, versus causation or causality, and where do you see that going? Is there any type of trend there on the literature right now?
I think most of the work produced right now, it's been based on association. So we know that by feeding certain additive, we see a change in the microbiome, but that doesn't really mean that we're actually targeting or tracing the specific input that is causing that change in the microbiome. So this is what we call association. Also, association is looking at the microbiome of different animals under different disease conditions. The diseased animals have a different microbiome compared to healthy animals. But again, this is an association experiment. To move to causation, there's different things that people are doing in the field. So the area of when you talk about human microbiomes, there's a lot of hype in using free germ mice, which is basically mice that have been devoid of microbes. So that way you can transfer different microbial communities to mice that are free germ and see if phenotypes can be reproduced on those mice that didn't have a microbiome previously. So that's when we become a little bit closer to causation rather than association, right? In the pig microbiome world, those words that relate to causation rather than association are still in infancy. There's some work being done in fecal transplants in piglets and also in some other food animals, for example, in dairy systems. And what we see in this work related to fecal transplants to replicate or reproduce phenotypes is that the microbiomes that's being transferred are difficult. It's difficult for those microbes to settle in the new system. That is, for a new microbe to colonize a new system, it has to first of all compete with the microbes that are already there and establish themselves so that the communities can be settled there in that system. Since we don't have a lot of opportunities to work with free germ pigs in the pig production area, those works and those experiments are very expensive. The work that has been done as far as looking at fecal transplants are, conventional pigs. And what we realize is that for that microbiome to be transferred to one pig that is high in production to a pig that is low in production and see if the phenotype can be transferred, I think we're very far away from that yet. Because we need those microbes to be settled in the new system to be able to cause some sort of change. So for me, the high promise would be on specific feed additives and looking at the effect of feed additives at the molecular level in pigs. So right now, I think people are doing work on moving from this compositional service of the microbiome to getting closer to the functionality of things. And the difference is that the techniques to study the microbiome in both cases are different. And of course, there's costs involved. There's also a lot of promise with using organelles. And this is something we're doing here at the University of Minnesota. We're growing organelles, specifically colonoids and enteroids in vitro. Mm-hmm. And treating those colonoids in those enteroids with specific microbial communities, but also with specific additives, and see if we can look at the changes in the organelle itself upon these stimuli with different microbes or different additives. That's a better way to get closer to causation, to get closer to the effect of specific additives or microbial communities in a very isolated part of the system, but that's a start. Looking at how microbes and feedstuffs interact in organoids to see if there's some changes in gene expression or changes in gut morphology that will help us get closer to probably what's happening in vivo.
Very interesting. Yeah, that's very cool research right there with those enteroids. Dr. Gomez, what is the biggest misconception about the swine gut microbiome?
I would say the biggest misconception. Okay, so I think people in the field are starting to learn more about microbes today. One of the misconceptions that we have in those studies, whenever people in the industry approach us to conduct different studies or to see whether a specific additive they have would have an effect in the microbiome, is that people sometimes are confuse the concept of composition versus function, right? So the typical, let's say, microbiome survey is based on answering the question, who's there? So what kind of microbes make up the system? So that basic microbiome survey does not really include the functions performed by those microbes. So if we want to know what the functions are, we will have to look for other techniques that would allow us to look at the metagenomic makeup of those microbial communities. So that's one of the misconceptions. People know that the typical microbiome survey would really reveal both composition and function, but that's not the case. To go to function, there needs to be additional analysis to be performed. Another misconception that people tend to have is that microbes are easily or can be easily manipulated, and that can be immediately translated to a phenotype of a desired phenotype of interest. That's a misconception because we're still far away from, let's say, managing to manipulate microbiomes specifically to twig or to have a phenotype of interest. That's still in infancy in the swine microbiome world. Those are probably one of the two misconceptions that I hear most when I interact with people that are novice in the area of microbiomes and want to perform microbiome work. Another thing is for designing an experiment in the microbiome field, as far as looking at the effect of different additives or different, let's say, drugs or any other compound that would have the promise to modify health or phenotype in pigs, is number of samples. We tend to look at very few number of samples in any given experiment, and what we've demonstrated that more than let's say the depth at which you look at the microbial system, the number of samples that you use in a given experiment, the power is very important. So looking at any kind of effect on any additive on pig performance, as in the context of the microbiome, is not going to be attained using 10 or 9 pigs per treatment. You need to look at many more pigs just to be able to get closer into that into looking at what specific effect has your additive into the microbial communities and see how that is being reproduced in the phenotype of the pig.
Very nice, especially the high variation that you mentioned, right? So that large sample size will definitely help there, right?
Yeah. So with large sample sizes, we guarantee that we are able at least to counteract some of that inter-individual variation. But as far as where we're going with the pig microbiome world, I think we still need to do a lot of fishing expeditions. So we call it fishing expeditions, but I would say hypothesis generating work. We still need to take a look at pig systems right from the beginning. So right now we're doing a lot of work in my lab looking at the piglet's microbiome before birth. So looking at what happened to the sow, what happened to different feeding strategies in the sow in gestation, and how that contributes to seeding the microbiome in the piglet at the very beginning. I think those surveys needs to be, or those experiments need to be done before you are able to devise any kind of feeding intervention or let's say additive intervention to be able to modify or to manipulate a specific phenotype in pigs. We need to know what happens with the seeding of the piglet microbiome right from the very beginning. And this is something we're doing very intensely here in my lab right now.
Very nice. Andres, what information or benefits can we get from gut microbiome research in the swine nutrition, health, and production fields?
I would say the biggest benefit. So the microbiome is an interface that, is the one part of the genome of the pig that we can modify immediately. You can do genetic improvement with pigs and look, after certain generations to see if you have improved a phenotype of interest. But the excitement about the microbiome is the one part of the genome of the pig that we can modify to see if we can obtain a desired phenotype. So I would say that the microbiome is the interface, for instance, between feeding and the genome. So whatever kind of feeding strategy that you implement on your pigs is going to be directly influenced by microbes. So that's one of the things that the people in the feeding industry can expect, in the feeding pig industry can expect about microbes. Any kind of feeding intervention needs to be or is intimately associated with what happens with the microbiome. So looking at, usually when you have any kind of, feeding trial in the swine nutrition field, you just feed the additive and you just see what happens with performance, right? You see if there was a weight gain, if there was feeding efficiency increased. But I would say by not looking at what happens at the microbiome, you're losing a piece of the picture. Because if you are able to trace what specific changes the microbiome experiences upon feeding certain, or specific feed additives, you can formulate a more targeted approach, especially if you know what specific microbes change. You're able to formulate a targeted approach to improve efficiency of the animal systems. So in other words, looking at changes in feed efficiency and weight gain upon feeding certain additives and not looking at the microbiome becomes a black box, right? Because you want to know the microbiome is the engine transforming that feed stuff into a bioavailable material that the pig is going to take advantage of. Of course, that depends on the nature of the feed additive, but I would say that's the most important piece of information that, let's say, pig nutritionists or swine nutritionists can get from microbiome work. What specific communities and functions change in function of the feeding additive that is being fed? And by looking at that, you're able to tweak, to adjust, let's say, the formulation of your feed or the composition of your feed to either enhance or boost or even inhibit the growth or the function of certain microbes to make this grow faster and more efficient. I would say that in a nutshell, that's what I'm most excited about when it comes to swine microbiome work. That by looking at these specific feeding strategies at the molecular level, we can know what specific members of the microbiome increase or decrease, and we can reformulate those feed additives in a way that can the desired performance or the desired effect or phenotype can be streamlined or can be obtained faster. But without looking at the microbiome, you're not gonna get that picture.
Very, very interesting, Andres. That sounds like a lot of work ahead of you.
Yeah. I mean, I think this is a very exciting time to work in microbiomes. I couldn't pick a better topic, to run a lab than microbiomes right now, because everybody wants to have the secret, what kind of specific manipulation can we, what kind of specific strategy can we implement to manipulate microbes and as a result, improve health and performance in animal systems. However, we need to remember that the microbiome is only a piece of the puzzle, right? So along with microbiome information, we also need information on functional genomics of the animal. We need information on nutrition. We need information on the metabolites that are being produced by the microbiome upon feeding certain food additive. So by looking at these questions in an integrated manner, I think people that work in the nutrition and the swine health field could have a better assessment of whether their additives or their let's say products or their any other kind of let's say product that is being formulated to improve health works, right? If you don't look at these different components of the system, microbes, functional genomics, metabolites, feeding, I would say you're missing a very important part of the puzzle, and I don't think you will be able to adjust and to tweak your formulation to make animals, healthier and more productive.
That makes sense. Any final words on microbiome, Andres, before we move to the 3 questions we ask every guest?
Well, I would say, my final word, I would say, the microbiome is a very exciting field of the animal and human health world right now. I would say that to move forward with plans about new feed additives, new management strategies, always take a look at the microbiome, see what the microbiome reveals. I would predict, Marcio, that in the future, in 10 years from now, microbiome is going to become PCR. That means every lab in any university or any lab in any company that, makes science or that, makes feedstuffs or any other kind of feed additive is going to have a microbiome station just to look at how microbiomes change or how microbial communities change upon feeding certain, feeding additives. So I would say my final word is for especially for people that work in the swine industry, especially when it comes to feedstuffs and nutrition, you got to take a look at the microbiome, because if you don't do that, you're missing a big piece of the puzzle.
Very nice. Celebrating its 25th anniversary, Gestal manufactures the original wireless standalone swine feeding system. Designed by pork producers for pork producers, they are simple, reliable, and provide peace of mind 24/7, 365 days a year. Gestal is not just manufactured by an equipment company, but by a family pork production business with a SLAT-level understanding. Gestal, always one step ahead in swine feeding. It is time to our famous 3. And there's going to the 3 questions we ask every guest, every episode. What is your favorite swine-related book?
Oh, well, my favorite swine-related book. Well, Marcio, my background, even though it's animal science, my background was never. I wasn't. Let's say a swine person in my early stages of training. My training was with non-human primates, and that's how I got connected to the microbial ecology world.
Or maybe another way to ask is your favorite gut-related book.
Okay, that's a better question. So I would say gastrointestinal microbiology is a and of course, it's written actually and edited by my advisor for my PhD in the University of Minnesota, Brian White and Rod Mackey. This is the Bible of microbiomes. It's the Bible of microbial ecology when it comes to gut microbiomes. It was written in the 1990s. Mm-hmm. It has 2 volumes, and it's called Gastrointestinal Microbiology. Okay. So this book was edited and written by the founding fathers of the microbial ecology world when it comes to gut microbiome. So anybody who's interested in microbial ecology, anybody who's interested in microbiome work when it comes to monogastrics or ruminants, needs to be aware of that book. It's called Gastrointestinal Microbiology. So it's not a swine book, but there's also a lot of chapters and sections dedicated to the swine gastrointestinal tract and its microbiome.
Very interesting. Yeah, I was not aware of that. So what is your favorite book unrelated to swine besides the gut books?
Of any topic? Any topic. Oh my God, that's I would say Malcolm X, the autobiography of Malcolm X. I don't know. Mm-hmm. I don't know if it has anything to do with microbiomes but for me, it's been a very inspiring book, not only from a personal but also from a professional standpoint. The autobiography of Malcolm X.
Very nice. Very good. And the last question, Andres, is what, in your opinion, sets apart successful professionals from those who are not?
Well, I can talk from my experience and also from my experience advising students. And what's made me Well, I don't know if I'm 100% successful. I would like to know. I would like to know that I am. But what's made me successful in this field, I would say, is the opportunity to experience science from different angles. So don't just focus on the specific narrow question that you have. Try to look at your question. Try to look at different angles of science to get a better assessment of the system that you're trying to understand. So, for example, in my career, I've been very fortunate of working with very, let's say, a wide range of seemingly very disparate people in different areas of science. I've worked with anthropologists, I've worked with psychologists, psychiatrists, people that work in the cancer field, people that work in the ecology field, architects, you name it. Very, very different and diverse range of scientists have allowed me to look at the problems that I deal with on an everyday basis from very different perspectives. So I guess my I would say what separates a successful professional in my field to other people is the willingness to listen to different opinions and to diversify the science that you do in many different fields. Those different assessments of science and to the problem you're addressing are going to help you deal and understand the questions that you have better. I would say that for me, it's what's made me successful in the field that I'm dealing with. And also, this is what I try to tell my students all the time. Whenever you have a chance to look at different problems, different questions, in a general way, I would take advantage of that.
Multidisciplinary approach. I love it. Very nice.
Exactly.
Very cool.
That's the word, multidisciplinary approach.
Well, thanks a lot, Dr. Gomez. Your time is much appreciated, and I'm sure you shed a lot of light on this subject for me and for the audience as well.
Oh, Marcio, thanks so much. I really enjoyed the conversation. It was a very fun conversation. I hope my insights on the microbiome world were of interest to the public, and I can be reached the University of Minnesota Animal Science Department in case there's any other specific questions about microbes, which is what we love to do around here.
Perfect. Thanks a lot, Andres. Have a good day.
Thank you, Marcio. You too. Bye-bye.